The overall goal of the following experiment is to visualize single receptors on the surface of living cells and analyze their location, motion, and dynamic association into supramolecular complexes. This is achieved by expressing SNAP-tagged receptors at very low levels on the surface of living cells and labeling them with bright organic fluorophores to permit single-molecule detection. As a second step, cells are visualized by total internal reflection fluorescence microscopy, which allows acquiring a fast image sequence of individual receptor particles moving on the cell surface. Next, automated detection and tracking algorithms are applied to the image sequence in order to obtain the position and intensity of each receptor particle over time. Results are obtained that show a precise analysis of the size of receptor complexes, in this example, beta-2 adrenergic receptors, based on a mixed Gaussian fitting of the intensity distribution of the particles at the beginning of the image sequence.
This method can help answer key questions in the cell biology field, such as how are receptors organized in nanodomains on the surface of living cells, how do they interact with each other to form dimers and oligomers, and how dynamic events at the basis of receptor signaling are actually taking place.
The main advantage of this technique over standard biochemical and microscopy methods is that it investigates the behavior of single receptors in their natural environment, thus allowing us to study important aspects which are typically hidden in ensemble measurements.